Let's review an important part of the innate immunity known as the complement system. Three major independent but overlapping pathways make up the complement system. The classical pathway, the lectin pathway, and the alternative pathway each have a distinct trigger, but all three pathways have the common result. They modify the target membrane by depositing C3 activation products and then engage a common terminal pathway called the membrane attack complex, or MAC, whose formation leads to cell lysis. The biochemical sequence for the classical pathway is initiated by the binding of C1q to an antigen-antibody complex on the surface of a pathogen or host cells. C1q then cleaves C1r, activating C1s, a serine protease complex. C1s then cleaves C4 and C2, creating C4b and C2a, which join to form the classical C3 convertase. The classical C3 convertase cleaves C3 to create the C3 activation products C3a and C3b. The lectin pathway is initiated by recognition of mannose residues on the cell surface by mannose-binding lectins, or MBLs. These molecules act through MBL-associated serine proteases, also known as MASPs, to cleave C2 and C4, again forming the classical C3 convertase and leading to the creation of C3 activation products. The alternative pathway is continuously active at a low level through spontaneous hydrolysis of C3, forming C3H2O. This process is termed tickover. Activated C3H2O binds to factor B, generating C3H2OB. Factor B is then cleaved by factor D and generates the alternative pathway C3 convertase. This enzyme catalyzes the cleavage of C3 molecules, generating activation products C3A and C3B. C3b also binds to factor B on cell surfaces, which is then cleaved by factor D to generate another alternative pathway C3 convertase, initiating the amplification loop. This complex is stabilized by factor P. Binding and cleavage of an additional C3 to C3 convertase forms the alternative pathway C5 convertase, which cleaves C5 to C5a and C5b. Factor P stabilizes this complex as well. The assembly of C5b with terminal components C6 through C9 forms the MAC on the membrane. The presence of soluble C3a and C5a also lead to coagulation, inflammation, platelet activation, leukocyte recruitment, and endothelial activation. To avoid self-amplification and self-damage, the complement pathway is tightly regulated. Dysregulation of the complement system is involved in a number of hematologic diseases, including paroxysmal nocturnal hemoglobinuria, or PNH. PNH is caused by acquired somatic mutations in the PIG-A gene in a subset of hemopoietic stem cells. Hemopoietic stem cells and their progeny that carry this mutation have reduced or absent glycosyl phosphatidylinositol, or GPI anchored proteins on the cell surface. This GPI anchor is essential for anchoring two complement regulatory proteins, CD55 and CD59, to the red blood cell membrane. These proteins protect normal red blood cells from complement-mediated lysis. In PNH, red blood cells that are GPI deficient do not have CD55 and CD59 on their outer membranes and are unshielded from attack by the complement system. The PNH red blood cells are unable to modulate complement activation on their surface, and the continuous spontaneous C3 tickover leads to surface complement activation with generation of alternative pathway C3 convertase. Once C5 convertases are generated, the terminal complement pathway is initiated, which eventually leads to MAC formation and MAC-mediated intravascular hemolysis.